This is Lecture 7 that goes with the cardiovascular system, or Unit 6. We have two major categories of immune responses in the body: the cell-mediated immunity, or CMI, and antibody-mediated immunity, or plasma-mediated immunity. Cell-mediated immunity is involved when the microbe is inside the cell or intracellular. Viral infections are always intracellular, and occasionally other types of microbes will be intracellular. The body is not able to remove the invader from the cell, so the entire cell must be killed. In antibody-mediated immunity, the invader is extracellular and not inside the cell. These can include bacteria, parasites, or fungi. The key factor in what determines which type of response you'll have is the invader inside or outside of the cell? An important principle in immunity is self-tolerance. The body must learn to recognize its own cells and proteins to avoid destroying them. The MHC is the marker that identifies self for self-tolerance. The immunoglobulins are immune proteins involved in the immune response that are in the plasma. When an invader is inside a cell, it is hidden from the immune response. The cell must display some foreign proteins in order for the immune system to react. The cell becomes an antigen-presenting cell when it processes the invader and displays part of the invader's protein on the surface to the other immune cells. It is so important not to kill our own cells, the immune system uses a co-stimulator to provide a second signal to confirm attack on the invader. Without the second signal to confirm the response, a foreign protein will not be attacked. For example, the TC, or cytotoxic T cell, will only be activated if it binds a receptor with the foreign antigen on the antigen-presenting cell, along with a second stimulus or co-stimulator. IL-2 is a common co-stimulator produced by the Th cells, or helper T cells. The Th cells release IL-2 not only to act as a co-stimulator to the cytotoxic T cells, but the IL-2 also attracts phagocytes to the area. It will trigger the B cells to do antibody production and to produce more plasma cells. The cytotoxic T cell will release enzymes to lyse cells, particularly the infected cells. It will also trigger the release of gamma interferon to activate the macrophages that have been called to the area. In the CMI response, the viral infected cell processes the invader and presents the antigen to be recognized. The cytotoxic T cell will kill the infected cell. Gamma interferon will be released to activate the macrophages to clean up the debris of the immune response. And there is a link that you can watch of an animation of cell-mediated immunity by Harvard Education. This link you can follow outside of this presentation. It is also going to be available within the links in Angel. Plasma cells are the clones of B cells that are produced to secrete one particular type of antibody in response to infection. A B cell is not able to produce enough antibody alone, so it clones itself to be able to produce more. The memory B cell is important after infection. Most of the plasma cells are recycled, but the body will retain some memory B cells which have memory of producing the antibody to that particular invader and will be able to respond more quickly and aggressively to any future attacks by the same invader. In the antibody-mediated response, the antibody is secreted to neutralize the antigen. If there are bacteria, the antibody will immobilize the bacteria. This triggers the antigen agglutination, where the antigen is coated with antibody or opsonized to be more attractive to phagocytes. Complement will be activated to increase the immune response and enhance phagocytosis. Again, there is another animation here you can watch on antibody diversity. It is available either through the Tegrity presentation or also on Angel separately. We have 5 categories of immunoglobulins. The first is IgG. This is present in the lymph and the intestines and is able to cross the placenta to the fetus. It is active against both bacteria and viruses. This will give a newborn infant a little bit of protection. IgA is present in secretions and is sometimes referred to as secretory IgA. It is present in sweat, tears, saliva, mucus, breast milk, and gastrointestinal secretions. It is active against both bacteria and viruses and is decreased during levels of stress. This will provide some additional protection to a breastfed infant that a formula-fed infant will not have. IgM is present in the lymph and is the first antibody secreted after initial exposure is detected by the body. IgD is present in the blood and lymph and is involved in B-cell activation. IgD is not well understood at this point, at least is not as well as the other antibodies. IgE is active in allergies, hypersensitivities, and parasitic worm infections. It works along with the mast cells and basophils in the allergic reaction.